consuming of the live microbial feedstock, or you can say the probiotics called light, mild and light type of digestive problem, but overall these are very safe for human to improve their health in a cost-effective manner, right? So it is always best to consult with a healthcare provider before starting any type of probiotics. Most common probiotics, according to the WHO and FAO approved, these are lactobacillus strains, bifidobacterian strains, and microbial, you can say yeast strains, right? Okay. Now, the global marketing, according to the probiotics, has been increasing day by day, and up to now, we have noted that the marketing of probiotics is by the end of 2027, maybe 85.4 billion by the end of 2027. So it's a very good business for any type of researcher or any type of scientist to just search out different types of microbial strains, and then just to use these strains and then prepare different types of food like baby food. You can say any type of food which can be improved the human health in very, you can say, natural and safe way as compared to by using the different types of antibiotics and other medicine. So it's a very good business. Probiotic products, these are very safe and very, you can say, emerging business all over the world. But the problem is that we have to use different types of, you can say, molecular method to just identify the probiotics, you can say probiotic identification. Like in past, there were different types of young scientists and researchers just use the 16S RNA gene sequencing to just identify the, you can say, probiotic strains from their research area. So it's not a valid and valid method, and some healthy issue, or you can say safety issues, will be emerging in the rest of the research. So in the second method, you can identify the probiotic strain with the help of in vitro and in vivo method, but again, the drawback is that it's very expensive and laborious method. Nowadays, machine learning is a powerful tool that can be used to search for, you can say, target-based probiotics. I will explain what is the probiotics and what is the target-based probiotics in my next presentation. But with the help of machine learning, you can easily identify the large data set of the microbial information, and then you can easily predict which microbe may be beneficial, which maybe have some beneficial, you can say, characteristics, or may have some beneficial properties for use as a probiotics, right? Before machine learning, you have to go for different types of steps, like you have to just search colony morphology of the bacteria, gram staining, catalytic test, oxidity test, you have to do fermentation, and then secondly, DNA extraction. Then you have to go for some PCR, gel electrophoresis, and then 16S RNA gene sequencing identification. And this the third step, you have to go for different types of probiotics, just like to just check the resistance of lower pH of that pre-identify probiotic strain, and then to have to just check the resistance of hemolysis activity, and then antimicrobial activity and antibiotic activity. Then in the step 4, then the genomics, again, you have to just build for a genomic sequencing. In the step number 5, you have just used that bacteria strain, and then just check the safety of that strain in the mice model. After all the hard work, you just have prepared your product for any type of research-based product, you can say, or you can say it's if it's for commercialization, it's depend upon or your on, you can say. But nowadays,
Dr. Shakira, you're muted.
You can see different types of machine learning based, you can say, tool. What is machine learning? It's a subfield of artificial intelligence that can be used, that use different types of algorithm and statistical modules to just enable your computer system to learn the data you provide. So machine learning is to just train your machine to recognize pattern and decision based on the data without requiring human involvement. In the human learning, we have just used intelligence. In machine learning, you use the model. In human learning, you just learn material, but in the machine learning, to just use the data. In the human learning, you just learning skill with help your own, you can say hands, but in machine will use all your data and then just give a, you can say, interpretation that your isolated strain is a probiotics or not. Okay, there are different types of, you can say, the machine learning method by identify the different types of, you can say, the target based. I will explain what is the target based probiotics and what is the generalized probiotics. Okay, what is different types of machine learning, you can say, types. Number one is the supervised learning, and in the second one is the unsupervised learning. In the supervised learning, we have to use the algorithm to train and to just label the data, and then in the unsupervised data, the algorithm is trained on the basis of data set and with no predetermined outcome of your label. I will explain in detail in the next presentation. You will be clear in that that slides as what are supervised learning and what is the unsupervised learning. Right now, machine learning in the searching of target-based probiotics. Now, what is the target-based probiotics and what is the generalized probiotics? So you have to think that in the target, in this simple probiotics, is a general probiotics. You use the bacterial strain and then prepare our general probiotics for human, for you can say baby food, for you can say it's a general probiotics. But in the target, you have to select what is your target. If your target is you just is your target is to just target the gastrointestinal health improvement, you have to identify the bacteria from the gut, and then you can just remove that you can say the bacteria strain from that environment and prepare that bacteria strain for preparation of your product, right? So the use of machine learning to just search the target gene. By identify these target genes, scientists can then use machine learning algorithm to search large number of microbes and identify those microbes that has only the desired or target characteristics. Another way of machine learning can be used is to analyze the metabolic pathway of the microbes and then identify those that are associated with the positive health outcome, right? This information, which we get from machine learning, can be then used to screen for potential target-based probiotics and then select these target-based probiotics for the preparation of any type of product. Overall, machine learning, therefore, to a very good and very powerful and very accurate means to search or to screen a target-based microbes from a huge data for preparation of target-based probiotics, and then to potential generally enhance the development of new probiotic product in the market. Okay. For example, for prediction of growth enhancer microbes from the genomic data, like you have to just enhance the growth parameter of poultry. You have to just search out the genes which will be only used for the preparation of the probiotic, which can improve the growth performance of the poultry, to improve the meat production. If your target is to just improve the milk production in animal, or to just control the colon cancer in human, you have to be very specific to select your target. By using the machine learning, you can easily identify, for example, if your target is growth, then you can easily identify the growth promoting gene and then the non-growth promoting gene with the help of machine learning in your computer data set. And you can also find high variable and non-growth bacterial factor in your microbial genomic data. Machine learning is a very any type of sequence genome that the growth enhancer. Okay. I think my screen is just sharing the previous slides. Let me check the problem here. Okay, machine learning in probiotics, right? In step number one, we have to extract, right? We have to just extract different types of feature from the genomic data. If you can say target is to just extract the growth promoting gene from your genomic data, we extract the feature from genome data, right? Then use these feature as a trained data set to train the selective model for prediction of the growth promoting bacterial gene identification. Right? So I am talking about the target just improve the growth of the animal health of a human. Then you have to just select those type of gene. So target based probiotics, these are referred to the probiotics that are specifically designed to target a specific health condition, or you can say area of the body. They are developed using molecular biology techniques to identify genetic traits, but the machine learning is a very good method to just screen out the target with the help of machine learning. It's a very good area you just identify with the help of machine learning. Okay. Um, so to the top objective is to development of machine learning based growth. You can say enhancer prediction bioinformatic tool, and the prediction of growth and available. As explained earlier, that the machine learning can be applied to predict, you can say the predict causing gene by using tool and this classification model. You can also build the page and web server for your own lab or company. Then the prediction, you can say the prediction of novel target-based predicts from whole genome, unknown genomic data. Number one, you have to just focus this screen and follow me step by step. You have to define what problem. As I also told you that if your target is to just improve the growth health, you have to select the data which from all over the world, Geneta database, right? NCBI, another genomic database, where just you can just take huge number of data. If your different types of researchers are working on, you can see the improve growth health. You can say if we just focus on the problem that growth and ungrowth related gene, if the growth of the animal is associated with the growth of the other animal, then you have to collect the data which is only those data which is having the capability to prove the growth of the animal. In step number one, you have to just collect the, you can say, literature, and then to just build a problem in your lab. Secondly, in second step, you have to just collect the genomic data. And if your research is probiotic, you have to collect genomic data according to the WHO and FAO guideline. And data can be taken from database, NCBI database, and it is DAR database. And genomic data, if you are working on, if I am working on my Pakistani healthy and unhealthy, you can say, poultry population, I will just collect the data from Pakistani environment and then put this data in my own, you can say, the library for building a machine learning based tool. So, but no, you have to just focus on that point. The data set should be, you can say, in a handsome amount to just build a good, you can say, machine learning based tool. And in my case, in my lab case, I just take almost 1,000 whole genome sequencing data from, you can say, NCBI and some data from my own lab. And the genomic size is from 1.5 to 8.1 and 1 mega based. Then you have to train your data set. You have to train your data set and independent data set. If your data is here, your data will be divided into two data set. One is a trained data set, and second is a negative data set, which is non-growth promoting, you can say, data. One is a positive data, and the second is negative data. In the positive data, you can have a gene which having the growth promoting enhancing gene, and in the negative data you have a no gene which having any type of link with the growth enhancing data. In this step number 3, you have to just use the feature extraction. You have to just pre-process, use different types of pre-processing method to train your data set. And for that, you have to use the, you can say, WHO and FAO guideline. What is your problem? It's your problem to just take those type of bacterial strain which can be used to in the lab to just prepare the poultry probiotics, for example, to improve the poultry health, or you can say poultry growth. Then you have to check the requirement according to the WHO and FAO, what their requirement. If they said that the Lactobacillus plantarum, Bacillus substrate, Bifidobacterium, Saccharomyces, and different types of yeast strains can be used to improve the growth of the animal, then you have to just extract the feature from these strains as compared to the randomly you're just going to extract non-growth promoting bacterial strains from the you can say research area. So step number 3, feature extraction is a very, you can say, crucial step to just build the pre-processing method, and then you have to just build your specific target to just train your data set. And in the step number 3, data set standardization by using different types of scale and algorithm. It's dependent upon your own research area. If your research area, from my example, is to just improve the health of poultry, and you have to just select those bacterial strain which having the gene which can be improve the poultry gut health, then you have to collect these strains and then train the strain to just negatively, you can say, push down those bacterial strain which having the no, you can say, bacterial capability to build the probiotics, right? In step number 4, you have to just select your own lab builds feature selection. If my lab is just working to select the bacterial strain having the improvement of the bacteria to survive in the gastrointestinal tract of the poultry, then I have to select that gene which having which is present inside the bacteria, which can use to improve the you can say control the gastrointestinal tract, and then it can easily survive inside the gastrointestinal tract of the poultry. So it's again depend upon your own problem. If the problem is in the poultry probiotics, you have to just select the data in the poultry. For example, if you are working on the cancer causing gene and non-cancer causing gene, you have to just target your own research area. If you're working on your colon cancer, you have to select those healthy, you can say, human gut in bacterial strains data which having the gene related to the anti-cancerous activity on the you can say colon cancer. And then on the other hand, you have to take the unhealthy patient, you can say colon cancer patient data, and then train your data set. 50-50 ratio, you have to train your data set. This is the probiotic strain which can cause, you can say, this is the you can say microbial strains which can cause the cancer, and these are the microbial strains which can't cause the cancer. So you have to train your machine and just split your data into two sets, and then at the end, while training your machine will only, by using the raw data, tell in the future that yes, your strain can be link have a link with cancer, and if its strain data set is very good, you can easily predict before going to any type of trial that yes, my machine give 100% accurate result, predict that this bacterial strain have no any type of cancer activity, and this bacterial strain has a cancer activity which can easily be diagnosed at early stages of the cancer. So again, it's depend upon your own research area. What's your research area? So back to my own lab course question, that I am working on the, you can say, the preparation of probiotic product to improve the growth health. So in step number 5, you have to train your data set into 80 to 20%. 80% you will train your you can say machine that this is a growth promoting bacterial strain, this is a growth promoting bacterial strain, and 20% is an active. And at the end, by using different types of state, rule, and scale and algorithm, you can easily evaluate your model, machine learning model, that whether it's a you can say a model is accurate or not. So again, it's a factors and it's a time consuming method to just take NCBI data. In this step number 7, after taking the huge data from the NCBI, and then after taking the, you can say, after extracting the feature selection, you have to split your data and then evaluate your data. And then by using the classification code and AUU, area and under curve method, you can also online your machine learning based model easily with the help of, you can say, the available, you can say, tools. So from step number 1 to step number 7, you can easily build your own machine learning based model for preparation of any type of, you can say, probiotics product based upon your own research. If your research is to just having a target that you have to build a anti-cancerous probiotic product to control the cancer. So you can easily search out the anti-cancerous, you can say, peptides from the genomic data, and you can build your you can say model, and then you can prepare your anti-cancer peptide probiotic product in your lab with the help of machine learning, right? Okay. Now comes to the result. After method, here the result. So there's a summary of the genomic data. You have to just take a huge data from, you can say, NCBI, whether it's a positive or negative, it's depend on lab. The same machine learning tool, if your target is to just build a target-based probiotic, you use different types of, you can say, program-based Python method to just build the machine learning model. So here is the, you can say, target-based probiotic product preparation enhancement tool. You have to take the data from NCBI and other, you can say, genomic bank. Then secondly, pre-processing of your data. And after that, you have to just split your data into 2 to 80 and you can say 20 ratio, and then model fitting and then model evaluation with the help of, you can say, the available tools. Right? Here's an example of our, you can say, on lab built model. Here we just use 1 to 7 step, and then we have just built a small, you can say, mini, you can say, tool to just predict whether it's a probiotic tool. You can also build your own machine learning based model in your lab if you have some specific problem. Here you can add some sequence. Also, you can add your own name with the help of different types available, you can say, software, your algorithm, and machine learning based, you can say, tools. You can also build your own web server in the lab. It's very easy, cost effective, and very accurate, you can say, tool. You can also write your lab and other things like if it's your target, you just filter out the huge data, whether it's a probiotics or not probiotics. You can just sequence and you can just build, and then you just paste your unknown sequence in you can say sequenced you can say block, and then after running that tool, your machine learning based tool will definitely predict that your unknown bacteria, yeast strain having some capability of probiotics or not. So it's a very accurate and you can say efficient tool. With the help of target-based probiotic, you can easily identify different types of marker, right? If you are, you can see, I have also told that this allow to the algorithm to learn to make the prediction based on input data. So in unsupervised, if in the type of machine learning, in unsupervised machine learning can be used to predict, you can say, probiotic prediction in variety of way. If one application is to just quality control, whether machine learning algorithm can, you can say, analyze product to data to detect defect and some abnormality in the production process, right? If any type of, you can say, missending in your data, the machine learning analyze the product data, any type of defect and abnormality on that. This can also help to identify the area production line you are working on colon cancer is working on. So you can have to just analyze the product data deficiency as well as the abnormality, and then prove the you can say product quality. And then defect if having low market you can say value, then you have to just stop that research and then just use those strains which have the high market value in the market. Like if you are just searching the growth promoting you can say bacteria strain, you have to search only those strains as compared to the low growth promoting bacteria strain. So this can also help to identify the you can say cash causing you can say probiotic. And another application of the machine learning it's also optimize the probiotic formulation. Market-based probiotic formulation is another application of the machine learning. By using machine learning, you can analyze the data, and then you can also predict the probiotic ingredient, and then the formulation, the protein content, the carbohydrate content, and the other you can say feed formulation, right? So by using machine learning to analyze data on product ingredient and formulation as well. So companies can identify unique idea. Machine learning can also identify the new combination of ingredient that can improve the product quality and then reduce the cost. So it's very good, you can say, area to just use machine learning to predict the future probiotics and then improve the probiotic quality and then reduce the cost. So machine learning can also be used to build the personalized or you can say target you can say based recommendation to individual outcome by analyzing the you can say consumer demand. By machine learning algorithm also used to you can say potentially generally enhance probiotic preparation by improving the quality control, optimizing the you can say formulation, and then you also personalizing the recommendation. And then overall machine learning has some very good potential to generally enhance the any type of probiotic product preparation. And the main thing why they improve the probiotic product preparation, they can improve the quality control. Number one, you have to control the quality. By using the machine learning, you can easily control the quality of any type of you can say product. Then you can easily optimize the product formulation. So it's very good area to just use the machine learning to search out different types of probiotic strain to prepare the probiotic product. And then the power of the probiotic, you can say machine learning, the company can also develop more efficient, more accurate, and effective production processes. And ultimately, they can deliver much better you can say feed, fodder, and food products, a unique area to just use the identify the probiotic strains and to just identify and novel probiotic strain to just give a efficient production process and then you just improve the quality control and deliver a better product to their consumer, right? So overall outcome of machine learning in microbiology, what is the overall outcome? You can provide commercial service of that machine learning tool as well. If your tool is very good, you can say it's you can also commercial you can commercialize your machine learning tool. I'm just repeating that by for building any type of machine learning tool, you have used just minimum cost. It's a very cost effective. You have just used the data from all over the world. So you can also provide commercial service of your own machine learning build tool in microbiology. In microbiology, the machine learning is very, the use of machine learning is very limited. Very, you can say, a huge gap is to just a link between machine learning and in microbiology. Although people are using the machine learning based tool to just you can say screen out and diagnose different types of cancer and other things. But the use of machine learning tool to just predict the probiotic based production is very limited. It's a huge gap. If you people just follow the 1 to 7 step how to build you can say tool, you can easily build different types of unique tool to just identify a unique probiotic bacterial strain from human gut, and then you can easily build your own tool, and you can provide, if your tool is unique, if having some unique ability, you can commercialize your tool, and then you can earn the money. Secondly, for student, you people can publish your paper in high impact factor journal. And if you are in some research and other things, you can produce number of cost effective target based, you can say probiotic in your lab, because with the help of machine learning, I am just, you can say again, you can easily identify different types of unique and very, you can say, you can say what I can use a word, very unique, improving quality control type of bacteria, and you can say but the unique improving health conditions probiotic strains will be, you can say, screen out, or you can say predict from machine with the help of machine learning, and you can build different types of probiotic product. If your working area is on plant, you can easily prepare the fertilizer to improve your plant health in very, you can say, safe and healthy manner. On the other hand, if your working area is human microbiology, you can build prediction, different types of, you can say, diagnostic tool to control the colon cancer, you control the breast cancer, to control the gastrointestinal cancer by taking the unique genes from the human, you can say, animal gut. You can prepare the cost effective and very safe, you can say, the product to control the cancer as well. But if you're working on agriculture sector, or you can say the livestock sector, you can also use the machine learning based tool to just screen out the health, or you can say meat enhancer gene from the animal gut, and then also you can screen out the milk enhancer gene from the high milk producing animals, and then you can use these bacteria strains to prepare the cost effective and very, you can say, healthy probiotic product to improve the animal health. So at the end, I am highly encouraged Dr. Wajia Ajmal, Scientific Officer, Bioinformatics Lab, Nigav, Pakistan, and also Mr. Yasser Akbar, Research Assistant, Microbial Bioinformatics Lab, NARC, Islamabad, Pakistan, for their input, for their, because the build for build any type of tool, it's you always need some team. So I'm highly, you can say, thankful to my team for just building different types of unique tool to just search out unique microbial, or you can say unique probiotic strains from a huge unknown data. And we are trying to just take a step further to just build new tool. And also we're just applying some copyright farm for our own build tool. Thank you so much. And if you have any question, I'm here to just answer you. Thank you so much.
Amazing presentation. That was powerful and superb. Really appreciate you for joining us. And I know very sure that a lot of people have really gained about this topic, especially now that the world is actually being transformed by the kind of revolutionary things that are going on in the industry and in the academic space using artificial intelligence and machine learning. So this is really a quite interesting area to explore. And I'm not very sure that a lot of people have a question. So guys, can you drop your question at, maybe you raise up your hand if you have a question, or you drop it at the chat room. We really appreciate it. Okay. So if you can drop your questions. I think we have a question here. I said that, can we use ML to make biosensor? Can we make ML to make biosensor?
Okay, I think there's a question for me.
Yeah,
Am
she
I
has a
right?
question for you. Yeah.
What type of question? Can you repeat again?
So can we have machine learning to make, can we use machine learning to make biosensor? I don't know if that makes sense to you.
Okay, okay. Yes, I understand. Okay. Yes, machine learning is a very powerful tool, yet that can be used to just assist in the search of different types of, you can say, material. Biosensor is also just to just sense the material to just build the unique, you can system to just improve the searching of the you can say machine. So why not we easily use the you can say data from the net and then build your own machine learning based tool to just build a unique biosensor for your own research area. So it's a huge, you can say, area. With the help of machine learning, you can build your own tool, whether it's a biosensor based tool, whether you can just build in, although I just talk about the machine learning use in the microbiology, but machine learning can also predict the early related, you can say the diagnosis cancer, you can say with the help of, you can say machine, you have to just train your machine that whether this is your positive control and this is your negative control is a, so only by you can say building a specific target, you just follow the same step, follow the different types of, you can say the formulas, and just build, you just I am just showing the steps here for preparation of any tool. Here, give me a few minutes, right here, here we are. You have to just focus that the backbone of the machine learning to feature selection, feature extraction. In the step number 3 and step number 4, I just mentioned that you have to select your feature. If your question is to just build a biosensor, yes, of course, you can have to just screen out the selection of your own research area. So it's totally depend upon the search area of your lab problem. What's the problem? So yes, of course, you can build the biosensor with the help of very, you can say, easy and accurate way. With the help of machine learning, you can build the biosensor. Yes, please. Any question?
Yes, so we have one more. You say, why use 80% positive data? It will increase the false
Okay,
positive
that's very
results.
question. Very good question. Right. So it's dependable. Okay, it's machine learning is in early stage. We have to just build. You can say a model. For model, different types of you can say the methods. And some you can say literature. 80 to 20% is the only way to just build your accurate model. You have to make sure that your model should be 80 to 90% plus in your you can say result at the end. You have to just build your own model, but 80 to 20% is not hard and fast to, but in my own case I have to just take 80 to 20% for building of, you can say, the machine learning model for own prediction of my own probiotic and non-probiotic. But scientists are working and they are giving different types of more, you can say, percentage area to just train the data set. So you have to train your machine. It's depend upon your own, again, your lab problems. What's your own problem? It's you are working on cancer causing gene, you have to, you can say, just improve that percentage well as well, and you have to take the, you can say, splitting formula for your own lab concern. Yes, please.
Amazing, amazing. Thank you so much. So someone asks again, can we use ML for drug target selection? Something, I don't know.
Drop silent.
Drug target selection, that means in drug discovery. Can we use machine learning to discover drug targets?
Yes. Drug discovery. That's very good question. That's the main role of machine learning is to just is to just give a huge information from a huge data, unknown data. We have a huge unknown data of a drug. We have to just select if our target to just take a anti-cancerous drug. You have a huge knowledge of your own research. Go to the literature and then select whether this type of antimicrobial or anti-cancer drugs can be used to build the anti-drug. You just trained your machine. And then for that, you have to take huge data from the, you can say, from your own lab, from the already published, you can say, data bank, and then train your machine. You need this drug from your own unknown data. So again, we can easily build a machine learning based model to just build a unique drug. Yes, by using these steps, you can easily, you can say, prepared a novel, or you can say the unique drug from the lab.
Amazing. Thank you so much. Uh, in bioinformatics, what group of ML algorithm is more appropriate? Supervised or non-supervised?
Okay, here is a question: is a supervised model or unsupervised model, right?
Which is
Here is a
appropriate?
question answer. Okay, in supervised model, I just explain in yes. Here is the question's answer. In the supervised model, you have to just the algorithm of machine learning is to train on a labeled data set, right? Labeled data set, where each data set or each data point is associated with specific label or outcome. So again, it's a your lab problem, your lab selection, feature selection. This in supervised learning model, machine learning model, the labeling will allow the algorithm to learn to make prediction based upon outcome data, input data. But in the unsupervised, where you have the unlabeled data set, there's no pre-prediction outcome and label. You just slow, if we just give your machine a specific type of algorithm and formula, you can easily build the, you can say, a model, but that model is unsupervised model. And if you just use the supervised or labeled database sets, you will just build your own supervised learning based model.
Amazing, amazing. Okay, so I think if we keep taking questions, we may not really leave this place. So I don't know if you can still take more questions. Are you okay with that?
Okay, I think no, I think I have to just prepare my Isha namaz. It's the time here in Pakistan.
All right, all right. So can you please give a kind of conclusion so that we can
Okay, but thank you so much. So I'm again repeating that machine learning is a very, you can say, powerful tool that can be used to just, you can say, assist in the search of for target-based probiotics. So again, I am just saying that with the help of machine learning, you can analyze the large data set of any type of microbial information, and then you can predict which microbe can be beneficial property for use of probiotic. So again, overall, machine learning has the potential to generalize, enhance the any type of probiotic, you can say, product preparation by improving the quality control, number one, quality control, and then optimizing the formulation, and then personalize the recommendation. So by using the machine learning, any type of company can develop very effective, or you can say more effective and efficient production process. And also, you can say, with the help of machine learning, you can easily deliver much better product to their, you can say, consumer. So use machine learning in your research. Use your laptop to just search machine learning based tool. How to just build machine learning based tool is a very easy way to just build machine learning based. If you have time to just learn the artificial intelligence based tool, because without machine learning, you will be no more in your research. So if you are a student, try to just learn what is machine learning. So learning is the only key to just you can just predict, or you can say, to search out unique, or you can say, novel product in your research. So thank you so much, and keep
thank you so much,
on
Dr.
the
Shakira. We
machine
really
learning.
appreciate you joining us. Thank you so much for lecturing us on machine learning in probiotics and generally in genomics. So we really appreciate that. We look forward to having you guys. So we have something we'd like to share very quickly before you guys go. We have a premium program that we'd like to introduce you guys to, just in case you like, you'd be interested. So permit me to share a program to those who are interested, before you go. So we'll be having another meeting next week, Saturday. So look forward to having you guys joining us. There's no certificate for this actually, because this was a meetup. So it's not necessarily a special program, okay? It's not necessarily a special program, so there is no certificate. It's just to educate life scientists. So we have bioinformatics workshops and research programs that we have in our company, and which includes building your career with us, gain practical research experience, and become an expert in your field of study. So our workshop is usually streamlined to empower life scientists with in-demand bioinformatics and computational biology expertise and promote competence in the biotech space. Our learning method practically engage students with critical thinking and innovative design to identify and solve biological research problems with live omics data. So we have programs like fundamentals of bioinformatics for those who will just be coming up to learn about bioinformatics. So if this is your first time of knowing about bioinformatics and you want to start your journey in bioinformatics and genomics, so this is an opportunity for you to have fundamentals of bioinformatics. I know some of you who are here with us already engage in this course, and you can testify how amazing this course is for you. So we have fundamentals of bioinformatics, and that's including introduction to bioinformatics and genomics, the construction of the use of software tools and internet tools, the diverse roles of bioinformatics in life science, ITS sequences, molecular identification of unknown sequence, retrieval of sample data from NCBI database, use of bioinformatics software for multiple sequence alignment, phylogenetic analysis of aligned ITS sequence, bacterial phylogenetic analysis at genome level, evolution and interpretation of biological tree. So it's like $40, but we're going to give it to you guys at a very discount price, at a giveaway price. So in case you're interested, it's your first time of knowing bioinformatics, can you drop on the group how much do you think we should give this away? How much can we give this to you guys? How much can we give you guys this? How much do you want to do? The current price is $40, but how much? If this is your first time to know fundamentals of bioinformatics, we have functional genomics too, where you deal with NGS data analysis, and then we have metagenomics analysis also, where you explore microbial NGS data samples that has been sequenced from NCBI database, big data samples, where you'll be able to explore AMR, virulence, drug target, visualizations of result taxonomy classifications, and the like. So we also have microbial genomics, where you explore bacterial things like probiotic. So we just learned on probiotics. So in probiotic, you learn how to do genome annotation, gene prediction, functional categorization, toxic compounds, adherence, stress response, vitamins, amino acid, immunomodulatory factors, antibiotic virulence factors, and so much. And also we have bacterial things like pathogenic bacteria that deals with genomics of infectious disease, where you're able to explore in detail antibiotic resistant genes, virulence factors, mobile gene elements, and so many things. And also variant sequence analysis, and plant genomics, therapeutic peptide. Currently, we have a 3-month metagenomics research program that deals with 3-month projects, and then you have a research paper that you can publish at the end of the day. So in case you're interested in this, it's just $100 right now instead of $300. And all these ones, we all have them at discount price. Instead of $100 right now, you can pay $50 for this. And then instead of $100 for this, you can pay $20 for metagenomics. And functional genomics is around, I think, $10 or $11 or so. Fundamentals also is at $6, I think. So in case you're interested, we are going to send the details on the group. So just join the group and enroll in the workshop. So and there's going to be like 5 days workshop each, and that is 5 days for the workshops and 3 months for the research program, in case you're interested. So can you drop on the chat room which of the program are you interested in? Anybody? Any case, so I believe so much that you guys are on the group, so we look forward to having you guys. Thank you for joining us today. See you next week. Bye.